Good morning. Now we will study one more important bacteria belonging to a group of diverse gram-positive bacteria called as actinomycetes. These actinomycetes are actually a transitional forms between bacteria and fungi. Means they morphologically they appear like fungi exhibiting mycelial network of branching filaments for which they were thought to be fungi. But later on the studies have revealed that they have a cell wall with muramic acid, prokaryotic nuclei, and they are susceptible to antibacterial antibiotics, which means they are actually bacteria. So they are true bacteria with a superficial resemblance to fungi, and they are related to mycobacteria and corynebacteria. These actinomycetes are gram-positive, non-motile, non-sporing, encapsulated filaments. Sometimes may appear as coccoid elements. They are free-living and they are present in soil. So basically they are environmental saprophytes. The genera among the actinomycetes belong to either aerobic genera or anaerobic genera. The most important genus is the Actinomyces, which is an anaerobic genus. The other anaerobic genera are Arachnia, Bifidobacterium and Rothia. The actinomycetes which are aerobic, that is the aerobic species, are found among the aerobic genera, which are NOCARDIA, Actinomadura, Dermatophilus and Streptomyces. Both of these actinomycetes, that is the members or the species belonging to actinomycetes, either aerobic genera or anaerobic genera, can cause a condition called as actinomycosis. Okay, so the clinical condition is called as actinomycosis, and the Streptomyces, which is a very rare cause of actinomycosis in humans, is a very important source of antibiotics like streptomycin. Now coming to the genus Actinomyces, as I told you, it is an anaerobic genus belonging to the group Actinomycetes. So before, the cattle used to exhibit a typical clinical condition or a disease called as lumpy jaw, wherein there used to be huge lump in the jaw of the cattle which exhibited multiple sinuses, and when examined the discharge from these sinuses showed mold like, means filamentous organisms, which was seen by Bollinger. Then the sinuses used to discharge granules which when studied or when observed under the microscope appeared like a ray fungus. Later on, the condition was described as actinomycosis, and the human lesions were discovered to be caused by Actinomyces israeli. And Actinomyces bovis is a common pathogen of humans. Now we have a large number of species belonging to the genus Actinomyces, the most important one being Actinomyces israeli. Others are Actinomyces odontolytica, Actinomyces naeslundi and others, which are commonly the normal flora of mouth, gut and vagina. Coming to the disease actinomycosis. It is a chronic granulomatous disease which is characterized by swellings which are indurated. These swellings are in the subcutaneous tissue, that is the connective tissue, and they show many discharging sinuses to the skin. These sinuses discharge pus or serous fluid containing granules, and there will be multiple sinuses in the swelling. Coming to the pathogenesis of actinomycosis. As I told you, invariably it is a endogenous infection because these are normal commensals in mouth, intestine and vagina. So then what could be the trigger? The trigger mostly is a trauma in the form of either dental extraction or surgery or the displacement of the intrauterine device or poor oral hygiene that may lead to entry of these organisms from their normal habitat of mouth, intestine or vagina into the subcutaneous tissue. The common organisms are Actinomyces israeli, naeslundi, odontolyticum, mayeri and others. The Infection of the face, that is which involves the actinomycetes obtained through mouth, invariably is enhanced by presence of other bacteria like Bifidobacterium, Actinobacillus, Echinella corridens, Haemophilus, Bacteroides and other anaerobic bacteria. All these bacteria, which are also normal flora of the mouth, provide an anaerobic environment for these actinomyces, which once enter into the subcutaneous tissue, grow in the anaerobic environment, leading to formation of indurated or swellings, multiple swellings, which have multiple sinuses pointing to the skin, discharging pus containing the granules. This condition is called as actinomycosis. Now, the clinical forms or the manifestations are cervicofacial, thoracic, abdominal or pelvic. Cervicofacial is the most common, or 60 to 70% cases of actinomyces present as cervicofacial actinomycosis. Here the infection is mostly endogenous from the mouth, presents as a indurated or hard swelling of the cheek or the jaw, exhibiting multiple sinuses. So there are indurated swellings and multiple sinuses. These sinuses discharge serous fluid or pus which contains the granules. Then the thoracic type or the thoracic actinomycosis is mostly a hematogenous spread to the pleura. The abdominal or the pelvic actinomycosis are due to either surgery, that is, as I told you, these are commensals in the intestine. So abdominal surgery may cause a spillover of this actinomyces from the intestine gut into the abdominal cavity, leading to abdominal actinomycosis. Or the pelvic actinomycosis is caused by displacement or dislodgement of the intrauterine device present in the uterus, which may lead to vaginal or pelvic actinomycosis. Other infections caused by actinomyces are gingivitis, periodontitis or sublingual plaques, that is the caries. Caries may be caused by actinomyces of the gut. And one more important infection caused by these actinomyces is the mycetoma. I will deal with mycetoma in the following slides. Coming to lab diagnosis of actinomycosis. So you people know the presentation of actinomycosis. Actinomycosis presents as indurated swelling or multiple abscesses which will have multiple sinuses discharging pus or serous fluid containing granules. Okay. So the specimen here would be either the pus or the serous fluid which shows or which has the granules, and in thoracic actinomycosis we will collect sputum for diagnosis. Then what is done with these granules? The granules are washed in water. Many a times we place a sterile gauze for collecting these granules. A sterile gauze is placed or it is sealed onto the actinomycotic lesion which has multiple sinuses, and it is left in place for over a period of 24 hours. Next day the gauze is taken. We can see a number of granules collected on the gauze. These granules are picked by the forceps and they are washed in water or saline, in a test tube containing saline, and then they are observed under microscope. What we see is in actinomycosis, yellow to white color, minute granules may be seen. They are observed under microscope by crushing between two slides, and when we do a gram stain we can see the gram positive filamentous bacteria. We can see gram positive center surrounded by a ray like fungal appearance, hence it was also called as ray fungus. When we put the same granules which are washed in saline, when we put them for culture, the media used is brain heart infusion agar for solid media and thioglycolate medium for liquid because these are anaerobic. Even the agar, that is brain-heart infusion agar, has to be incubated in anaerobic conditions. In the liquid medium, we see that fluffy ball-like growth is seen after 2 to 3 days in thioglycolate medium, and brain-heart infusion agar, when inoculated with these granules and incubated anaerobically, they take around 7 to 10 days to grow into large colonies. These colonies initially appear spider-leg colonies, the colonies which have multiple tentacles like a spider leg, and later on, within a period of 7 to 10 days, they become large heaped up colonies. The species identification is further done by biochemical reactions, fluorescent specific antibody methods, or HPLC or GLC method, and immunofluorescence. This disease, actinomycosis, is mainly seen among the rural area and men who are exposed to the agricultural work, that is, who are exposed to vegetation, who are exposed to moist hay, or the hay after rain which becomes mushy will have large amount of this mycelial bacteria. So when the farmers or the agricultural people inhale this, they get actinomycosis. So that is why cervicofacial is most common actinomycosis seen. Abdominal and pelvic, as I told you, the predisposing factors are either a surgery or dislodgement of the intrauterine device. Treatment is with penicillin in people who are sensitive to penicillin, but a long term treatment of 6 to 12 months with penicillin is recommended. People who are allergic to penicillin, tetracyclines are preferred. Simultaneously, debridement surgery may also be done if the tumor or the swelling is very, very large. Now coming to the aerobic genera, the aerobic genus NOCARDIA. NOCARDIA are also actinomycetes which are aerobic, gram positive. They are mainly present in soil, so they are soil saprophytes. Important species are NOCARDIA asteroides, NOCARDIA brasiliensis, and NOCARDIA cavea, which can cause human infections, and they cause cutaneous, subcutaneous, or systemic infections. The cutaneous and subcutaneous infections are mainly caused by inoculation of these mycelial elements into the skin when the farmers walk barefoot. And the systemic and metastatic lesions are mainly caused by inhalation of these mycelial structures by these people who professionally get exposed to them. Coming to diagnosis of nocardiosis, the condition is called as nocardiosis. So if it is a thoracic or respiratory infection, generally we collect sputum, and the sputum, when we do a gram stain, we can see branching gram positive filaments present in the sputum, which will give a clue towards presence of actinomycetes. And the growth is again on brain-heart infusion agar. Here we can see wrinkled granular colonies, and the treatment for nocardiosis is cotrimoxazole. In immunocompromised, we prefer aminoglycosides like amikacin. Now coming to mycetoma. Mycetoma is a chronic granulomatous condition of subcutaneous tissue characterized by swelling, multiple sinuses, and these sinuses discharge granules, and this mycetoma, this condition has a risk of causing or spreading to the adjacent bone, leading to bony irregularities. This mycetoma can be caused by either fungi or bacteria. When caused by fungus, it is called as eumycotic mycetoma, and when caused by bacteria, it is called as actinomycotic mycetoma, because actinomycetes are the bacteria which can cause this mycetoma. So actinomycotic mycetoma is mycetoma means localized granulomatous subcutaneous or deep lesion of foot or hands. Hands are also involved, though rarely, and it presents with a swelling and multiple discharging sinuses caused by actinomycetes the first actinomycete to have been discovered or associated with mycetoma is the Actinomadura madure, hence it was called as Madura mycosis. Sometimes we can see similar clinical picture produced by bacteria as well, other bacteria as well apart from actinomycetes. So, the clinical picture is like that of mycetoma, means there is a swelling, there are multiple discharging sinuses, there may be bony deformities and these sinuses discharge granules, but there may be no or absolutely no actinomycetes. The other bacteria which are associated with a similar condition is Staphylococcus aureus, and the condition is not called as mycetoma, it is called as Botryomycosis. Okay. So, the bacteria causing actinomycotic mycetoma are Actinomyces israeli, Bovis, Nocardia asteroides, Braziliensis and KVA, Actinomadura madure and Actinomadure pelletieri. These are the actinomycetes which can cause mycetoma, and Botryomycosis is a clinical condition which resembles mycetoma but caused by other bacteria like Staphylococcus aureus. Okay. So, the diagnosis of actinomycotic mycetoma is based on the studying the granules. The granules of actinomycotic mycetoma are mostly white to yellow, whereas black granules are seen if it is a purely fungal, means eumycotic mycetoma. Rest of the diagnosis remains same, that is the granules are observed under microscope, then they are put up for culture on brain heart infusion agar. These actinomycetes can also cause hypersensitivity reaction. I told you these actinomycetes are environmental saprophytes, they are present in soil and vegetative matter. So, people who come in contact with this vegetative matter or who inhale these mycelial forms may have a hypersensitivity or they may exhibit a hypersensitivity reaction, and the actinomycetes which commonly causes hypersensitivity reactions are those belonging to the genus Phania or Saccharomycosporia, and they result in a pneumonia like condition which becomes chronic obstructive pulmonary disease. This is also called as farmer's lung. So, to summarize, you have studied today the bacteria belonging to group Actinomycetes. So, the Actinomycetes can be either aerobic or anaerobic genera. The anaerobic genera are more common. Actinomyces is the genus which is more commonly causing infection in humans. The aerobic genera, members of the aerobic genera are mainly Nocardia. So, what infections they can cause? The Actinomyces, the infection caused by Actinomyces is called as actinomycosis, and the common presentation are, as I told you, cervicofacial, which is most common, then is the thoracic or pleural, then abdominal and pelvic. In all of these we can see a similar presentation, that is a tumor or a swelling will be present with multiple discharging sinuses which discharge granules, pus or serous fluid containing granules. So, the specimen for lab diagnosis would be either the granules or sputum in cases of thoracic actinomycosis, and microscopy will reveal that they are gram positive filamentous bacteria seen. The histopathology may show a ray fungus, that is in the center there will be large eosinophilic filamentous structures surrounded by basophilic club like structures, which the appearance is described as ray like fungus. Then the growth of these organisms can be done anaerobically if you are suspecting actinomyces, and the media used are brain heart infusion agar and thioglycolate liquid medium. In both of them it takes around, in liquid media around 2 to 3 days it grows, and solid media takes for a complete growth to appear around 7 to 10 days, and the treatment of actinomycosis is penicillin. People who are sensitive to penicillin is given over a period of 6 to 12 months. Those allergic to penicillin, we prefer tetracyclines. Then the aerobic genus, the members belonging to the genus Nocardia are most common. So, Nocardia asteroides, Nocardia Braziliensis and KVA are the most important species causing nocardiosis. The diagnosis and treatment remains same. And actinomycetes can also cause what we call as mycetoma and hypersensitivity pneumonitis called as farmer's lung. Thank you.